onto the small intestines we go. Chyme has just left the stomach. We've entered the small intestines, and granted, in the previous video we've talked just a teeny bit about as that duodenum, the first segment of the small intestine, starts to distend, it initiates a reflexive response back to the stomach, okay, to shut off gastric secretion, okay. And so, we alluded a little bit to some of the early functions of the small intestines. So when we look at the gross anatomy of the small intestines, it's actually broken down into 3 subdivisions. We have the duodenum, or duodenum, the jejunum, okay, and the ileum. Again, how you learn the terminology is all based on the person and where they're located from. So a lot of British kind of training will call this the duodenum. I've always called it the duodenum. But again, so the terminology might be slightly different based on who you've heard it from. So this is the major organ. The small intestine is the major organ where digestion and absorption is going to occur. The bulk of your digestion and the bulk of your absorption are going to occur in the small intestines. It ranges anywhere from 2 to 4 meters long, so it's a fairly long organ. And it runs from the pyloric sphincter, which separates the stomach and the duodenum, and the ileocecal valve, which separates the ileum from the cecum of the large intestines. The small intestines is located essentially from here. Oh. So here's where the ileocecal valve would be, and here is where the pyloric sphincter would be. So this entire structure sneaks behind the large intestines here. All of this structure inside of here, all of this here is the small intestines. So when we look at the 3 different divisions, the duodenum, again, is the shortest of the sections, and it's basically just this initial little curvature region. It surrounds the pancreas, and actually has a large role or connection to not only the liver and the gallbladder, but the pancreas as well. And that's because the ducts of both the gallbladder and the pancreas empty into the duodenum. And we'll talk about this here in a little while and the importance of these 2 ducts and their role in the digestive process. As that chyme moves on through the duodenum, it then enters into the jejunum. And the jejunum will extend from the duodenum to the ileum. It is around 2.6 meters long, where it joins the ileum, and the ileum will then join the large intestines. Just like we talked about some modifications in the stomach, there's some significant modifications in the small intestines. I've alluded to the fact that the small intestines is the major area for not only digestion but absorption. So if we have a massive amount of absorption we need to increase the surface area of this organ. And it does this kind of through a number of mechanisms. One, it does it through circular folds. So circular folds, so as you can see, this is a tubular structure that's coming down. And what you can see is winding up through this tubular structure are these folds, okay, along this structure. And so as you can envision, if you had just a tube, right, with smooth edges, that solution would just go down past those edges. However, what the junctional folds do is they basically cause the chyme, as it's entering the stomach, right, to basically swirl around the edges as it goes down through the structure. I always think about this, and the major place where I see these big contraptions are at children's hospitals for some reason. Like, it's like the mousetrap mazes, right, where you have the ball that gets clicked up to the top, and then it gets released, and it goes down that spiral, right, and then hits something else, and blah, 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 okay? So, again, that ball could just shoot down, right, would move really fast, or it can spiral around the edge, which takes it more time, and that ball basically is then exposed to more surface area. That's exactly what these circular folds are doing. They're increasing the amount of surface area that chyme is exposed to so that we can increase nutrient absorption, okay, within the small intestines. Now, if you zoom in on these circular folds, what you will see is the presence of these villi. So here is the epithelial kind of surface down here, and what you can see is these huge projections basically extending out into the mucus, or out into the lumen coming off of the mucous membrane. And inside of these villi, or these villus or villi, what we see is a really nice capillary and lacteal, which is, a lacteal is really part of the lymphatic vessel system. Okay. And those capillaries and that lacteal is required, or really important for the absorption process, right? Because once we absorb the nutrients out of the small intestines, we have to be able to get it somewhere else, right? And that somewhere else is our bloodstream and the lacteal of the lymphatic system. Now, if we zoom in even more on these villus, what we will find is that the epithelium that exists on this villus have microvilli. Now, you're going to hear me refer to later on brush border cells. These are the brush border cells. So these microvilli exist on brush border cells. Okay. And these brush border cells, or these microvilli, are going to basically contain enzymes for carbohydrate and protein digestion. So the surface of them will carry out digestive processes. Okay. Now, we have intestinal crypts within the epithelium that are renewed every 2 to 4 days. Most are basically secretory cells that secrete intestinal juices, which I'll talk about here on the next slide. They do have a large population of stem cells that divide to basically replace the cells of these crypts. Okay? So one homeostatic imbalance that we often see, or that we see, is when patients undergo chemotherapy treatment. Most chemotherapy treatments target dividing cells. Okay? Excuse me. One of the hallmark characteristics of cancer is that they divide uncontrollably. Okay? So if a cell is dividing uncontrollably and you can prevent it from dividing, then it becomes a great target for chemotherapy agents. The majority of the cells in the body sit in something called quiescence, which means they're not dividing, they're not doing anything crazy. Okay? However, there is a small subset of population of cells in your body. We talked about the stomach. Now we're going to talk about the intestines, or the small intestines, where you have this very rapid turnover of cells, okay, as a protective mechanism. Okay. So unfortunately, even though chemotherapy targets and kills cancer cells, it also kills the rapidly dividing cells of your gastrointestinal tract. This is why the chemotherapy, or a lot of chemotherapies, result in this nausea, vomiting, and diarrhea, because of the fact that you're setting up this homeostatic imbalance within the digestive system, and that response, or that homeostatic imbalance, creates these symptoms. So the intestinal juice that was referred to previously on 2 slides ago, is the juice really, it's coming from the cells of the small intestine. We produce about 1 to 2 liters of this a day. And it's going to be released in response to distension, the stretching, as well as irritation of the mucosa from that kind coming from the stomach and entering into the duodenum of the small intestines. It's slightly alkaline. It tends to be isotonic with blood plasma. Again, isotonic meaning the same ionic makeup, or the same tone as our blood plasma. It's mainly water. It doesn't really have any enzymes associated with it. Now you might be like, well, wait a minute. Like, you just said a little while ago that digestion is a large part of the small intestines. It is, but those digestive enzymes don't come from the intestinal juices that are being created by the small intestines. They're going to come from the liver, the gallbladder, and the pancreas, which we're going to talk about here shortly. So the intestinal juices also facilitate transport and absorption of the nutrients. And so really the intestinal juices are kind of like a vehicle. They're just going to kind of dilute out the chyme a little bit. They're going to serve as a little bit of a protection. They're going to help facilitate the movement of these ions out of the small intestines. Okay, but it isn't involved really in any digestive component or mechanism of the chemical digestive process. So we're going to move into talking a little bit about some of our accessory structures of the digestive system. So the first accessory structure is the liver and gallbladder, and these are typically paired together because they actually have a functional relationship to one another. So the liver actually has a huge number of functions within the body. Only one of those functions of the many hundred is actually a digestive function, and that one function is the production of bile. So the liver is where the bile is produced. Okay, it is then sent to the gallbladder where it is stored. Okay, so what will happen in the gallbladder is a lot of the liquid is removed, and so what you get is just this really highly concentrated bile. Now, what bile is a fat emulsifier. So imagine a fat blob that looks like this, right? And if you had a whole bunch of enzymes trying to eat away at the surface of this, or trying to break this down, right? It would be really hard to be able to get to the inside of this globular material. So what fat emulsifiers do is they break this big old fat molecule down into smaller little droplets. Okay? And what it does essentially by doing that, right? Now these enzymes can break down much easier, right? These little teeny microscopic droplets versus a big old blob, right? You can get those enzymes into those little teeny, they don't stay confined touching like this, they basically break them down. So you have these individual pieces over here like this, right, versus this big blob. Okay, and now the lipases can come in and target these much easier than this huge, big, massive lipid. Okay, so that's what a fat emulsifier does. Let me give you another kind of visual example. So you take oil and vinegar, right, salad dressing, and you put it down, and you can see the oil and the vinegar separated. You shake it up, right? It looks like they're all combined. You sit it down, and they separate back out. Okay, and that's because lipids and water, vinegar is a water base, don't like each other. Now, if you were to put an emulsifier in that oil and vinegar and shake it really, really, really well and sit it on the counter, you would not get that separation. Okay, because you've created such microscopic droplets of fat that doesn't have that big, bulky, repelling component. Now, if you left it for days and days and days, you probably would start to see a little bit of lipid layer form. And then over a long period of time, you'd probably get that separation back out. Okay? But again, it's there to break down that larger fat molecule into a much smaller, manageable molecule or size so that it can be digested later on. So bile tends to be a yellow-green alkaline solution containing bile salts, which is a derivative of cholesterol. Again, this bile salt acts as the fat emulsifier. It also contains bilirubin. This is a pigment from heme. It is also the pigment that gives our feces its brown color. So what happens is the bilirubin enters our digestive system, or not enters our digestive system, enters the large intestines of our digestive system, and there the bacteria of our large intestines actually start to break down the bilirubin into something called stercobilin, and this is what gives the feces its brown color. Okay. In addition to those two major components, you also have a little bit of cholesterol, some neutral fats, and phospholipids, as well as electrolytes contained in our bile. So the gallbladder itself, again, is this teeny little structure that exists on the superior aspect of the liver. It stores and concentrates bile by absorbing water in ions. So essentially the bile from the liver is sent to the gallbladder, where the water and ions are extracted out, leaving the bile salts and the bilirubin and some of the other components in the gallbladder to concentrate. Eventually there'll be a signal and a single event muscular contraction will cause that concentrated bile to leave the gallbladder through, the cystic duct and then down into the bile duct, which will then empty into the duodenum. Now, what happens sometimes if you have really high cholesterol, what happens is you get too few bile salts being created, and as a consequence of that, your kidney, or not, oh my gosh, why do I keep on saying kidney? Your gallbladder will actually form something called gallstones. And these gallstones they're similar to our kidney stones we get in our kidneys. But essentially what happens is you start to build up these little stones in here, and as you can envision, as we build up, little stones, if we have these little stones that are building up here, you get this muscle contraction, one of these stones goes out, and boop, it occludes or obstructs the flow of bile out of that gallbladder. And so what ends up happening is obviously this becomes super painful. You get a buildup of fluid behind here. You can have, there are medications as well as different treatments to actually address these stones that are formed. And some people actually have to have their gallbladder removed. Now, if someone has a gallbladder removed, they end up, what that means in terms of digestion is that they can't digest fats as well. They still be able to digest some, because we're going to learn here in a minute that when we want to release this bile, the bile that's in the liver is also going to be secreted. So you have this dilute bile coming out from the liver, this really concentrated bile coming from the gallbladder. So if you remove the gallbladder, you still have a little bit of bile coming down. It's just not going to be enough to be able to carry out digestion of all of the lipids in your diet. So two homeostatic imbalances that we see, and they're kind of not necessarily two homeostatic imbalances, we see these kind of one leads to another. Within the liver is something called hepatitis. Again, there's our inflammation of the liver. Hepatitis can be seen due to viral infections, so hep A, B, and C, all target, so hepatitis A, hepatitis B, and hepatitis C, all target the liver. Drug toxicity can result in hepatitis. So for example, excess alcohol consumption can lead to hepatitis. Wild mushroom poisoning can also lead to hepatitis. Now, if hepatitis becomes chronic, and persistent, this will often lead to cirrhosis. And cirrhosis is often something I think we most classically associated with people who are alcoholics. And in cirrhosis, what happens is the liver is actually replaced with fatty fibrous tissue. So this is what a cirrhotic liver would look like. And this will often lead to portal hypertension. So a person will have hypertension, that high blood pressure. It's a secondary hypertension because it's due to an underlying cause. If you address the liver condition, then you resolve the hypertension. Unfortunately, the liver cannot replace itself, cannot regenerate, and a transplant can be very effective. However, livers are super, super, super hard to come by for some reason. And so oftentimes people will end up dying waiting for a liver. Okay. Now, when I say the liver does not regenerate, the liver cannot, after being damaged, it cannot regenerate. Okay. So in this case, this is organ-wide damage. And I bring that up because there is a little bit of a caveat. So the liver is a unique structure. if you took a healthy liver, okay, so let's pretend like this is our healthy liver, okay, and I cut out and I removed just this portion, okay, and I transplanted that little portion, right, this little portion here, into a person. Over time, that portion would grow into a normal sized liver. So the liver does have the ability to regenerate. It does not have the ability to repair once it's damaged, I guess is a better way of putting it. Okay, so the liver is a unique organ like that. Okay, our other accessory structure we're going to talk about is the pancreas. So the pancreas, we've talked about the pancreas in the endocrine system. We talked about its role in its ability to secrete insulin and glucagon, and that's its endocrine function. It also has an exocrine function. Now again, remember the difference between these two is endocrine glands secrete into blood typically, okay? Exocrine secrete are glands that have ducts that secrete onto the surface of something. If we look at our pancreas, here is our duct, and this is the duodenum. The mucosa of the duodenum is the surface that we're referring to, okay? So there is, the pancreas is unique in that it has both endocrine and exocrine function, okay? In terms of the digestive, we slowly rely on the exocrine function in terms of its digestive processes, okay? Now, when we talk about what the pancreas is going to do, it is going to produce pancreatic juice. Crazy, right? Pancreas creating pancreatic juices. However, this is like the rock star of our digestive process in the small intestines. Okay, we produce about 1,200 to 1,500 mils of this a day, so 1.2 to 1.5 liters of this a day. It has a large, or it's slightly alkaline, so pH of 8, but it has a large amount of bicarbonate. And the reason for this is this is going to neutralize that acidic chyme as it enters into the duodenum. But more importantly, what we can see is that this pancreatic juice is comprised of essentially every enzyme we need to carry out all of our digestive processes. So we have our amylases, right? These are going to target our carbs. We have our lipases. These are going to target our lipids. We have our nucleases. These are going to target our DNA, okay? And we have our proteases, which will target our proteins. Now, your amylases, lipases, and nucleases, those are enzymes. Again, here you can see that ACE, right? ACE, right? I told you if you ends in ACE, it's typically an enzyme, okay? These three classes of enzymes, amylase, lipase, nucleases, are all secreted from the pancreas in their absolute active form, okay? However, our proteases are secreted in their inactive form. That means that once they hit the duodenum, they still have to be activated. Now, there's this whole complex cascade of how they become activated, okay? All I want you to realize or to know is that our proteases are released inactive, okay? The major enzyme being trypsinogen. And when that becomes activated, it turns into trypsin. And it does that by the brush border cells, those enzymes I told you about already on the when we talked about the villus, the anatomical structure. And this trypsin activates all other proteases. Okay? Don't worry about the names of all of these other proteases. They're inactive or their active form. So I don't really want you to realize, you don't need to memorize these, but I do want you to, and the reason for this is if you ever, trypsin and trypsinogen are very common, especially in a research environment, in a clinical environment, they talk a lot about trypsin and trypsinogen. more importantly, trypsin as something that will break down proteins. Okay. So again, you can kind of ignore these names and just kind of replace that with all other proteases. Okay. Now, just like the stomach has a regulatory process of triggering the secretion of those gastric juices, which we talked about a few videos ago, the small intestines also has a process, a series of hormones that will trigger the release of not only bile, but those pancreatic juices. Okay. So I want you to go back and think about that intestinal phase of our gastric secretion. Remember in that intestinal phase, and let me just go back. Oh, I think I just went back. So here's that intestinal phase, right? This inhibitory, right, where we secrete secretin and cholecystokinin, okay? And these go back and target the stomach, okay? These two hormones that are secreted, okay, are the exact same hormones that are secreted here, okay? When we talked about it in the gastric phase, chyme enters, right? We get distension, CCK, and secretin, right? Go back and shut off the stomach, okay? So they're actually doing two things. They're shutting off the stomach, but they're turning on the secretions of the duodenum, okay? So in our duodenum, we have a population of enteroendocrine cells as well. So the stomach has enteroendocrine cells, the duodenum has enteroendocrine cells, okay? This is the population that's secreting that cholecystokinin or CCK and secretin, okay? So that CCK and that secretin enter the bloodstream. And the intestinal phase of gastric secretion enters the bloodstream and shuts down digestion in the stomach. Meanwhile, as that chyme continues to enter into the duodenum, okay, it promotes our accessory organ structure secretion. So what happens is the bloodstream, okay, CCK, secretin enter the bloodstream, and both of them, more CCK than secretin, causes the secretion of our pancreatic juice. Okay, so CCK and secretin target the pancreas, promote secretion of the pancreatic juice into the duodenum. That CCK goes on and targets the liver or targets the gallbladder. And CCK, only CCK in this effect, causes that muscular contraction of the gallbladder and the release of bile into the duodenum. CCK and secretin target the liver, cause the liver to secrete the bile. Okay. And now we have our chyme with all of the enzymes coming in from the pancreas, with the emulsifier coming in from the gallbladder as well as the liver. Okay, so now we have this duodenum consisting of all the enzymes we need. The proteases are becoming activated. Our chyme is there, and our food particles are ready to be broken down. So when we look at the digestive process in the small intestines, chyme from the stomach contains partially digested carbohydrates, which are being broken down by that mechanical and just the pH of the stomach acid to begin with, as well as partly digested proteins. But our fat is pretty much undigested. So that chyme will basically travel through the small intestines from anywhere from 3 to 6 hours. Okay, in that time frame, almost every single nutrient is going to be absorbed through the small intestines, and the majority of your water will be absorbed. So again, there's a massive amount of absorption through the small intestines in this process. And again, if you go back and look at the modifications we talked about earlier, this is why we have those modifications. Okay. Now, in order for us to be able to carry out all the digestive and absorption in the small intestines, we need a few things to happen. We need a slow delivery of that chyme. Like, it can't just be dumping the whole thing in at once. Okay? We need to make sure that the bile and the enzymes and the bicarbonate ions from the eye in the liver mix with our chyme and neutralize it, because the enzymes here are not active in an acidic environment. So if we don't neutralize this chyme with this bicarbonate ion, we destroy these enzymes and we cannot carry out digestion. Okay? We need mixing to occur, right? It doesn't help to dump all this in and have our chyme over here and there's no mixing occurring. So we need segmentation to occur. Okay? If you don't remember what segmentation is, go back to like the 4th or 5th slide of this chapter and you'll see what segmentation is. It's the most kind of motion in the small intestines, and it mixes and moves contents towards the ileocecal valve. Again, this is a fairly slow process. The last thing that we see as a requirement is peristalsis. Okay? This is initiated by the rise in a hormone called motilin, and this occurs late in our intestinal phase, and it occurs about every 90 to 120 minutes, right? So this is about every hour and a half to every 2 hours. And it's a wave that occurs from the duodenum and travels towards the ileum. Now, what happens is with each wave, okay, you move that food product closer and closer and closer to the large intestines. Now, this movement can all be accomplished in about 2 hours. But again, this isn't triggered, okay, right when the chyme enters into the duodenum. There's a slight delay until we get enough motilin. So you can have 4 hours of digestion before peristalsis kicks in. Once peristalsis kicks in, you're going to move all of those contents into the small intestine or into the large intestines within about 2 hours. Okay, so that breaks down the small intestines. In the next video, we will talk about the large intestines.